Hello, good afternoon, good evening. Thanks for being here. A very warm welcome to today's webinar, The Use of Organic Acids in Poultry. My name is Arun and I am the Marketing Manager with BIOMIN in India. I will chair and moderate these sessions for you today. For today's sessions, we have brought in three experts, Dr. Neil, Dr. Gangga and Dr. Selvam, all of them my colleagues from BIOMIN. We will now hear a brief self-introduction from these in-house experts. Dr. Neil, may I ask you to introduce yourself first, followed by Gangga and Selvam, please. Thank you.
Thank you, Arun. Good afternoon, ladies and gentlemen. It's a pleasure to be here with you. As you can see, my name is Neil Gannon. I'm the Regional Product Manager for Gut Performance. I've been with BIOMIN for nearly 10 years now, and for the last 5 years in this role as Gut Performance Manager, in which I support our customers and sales staff across the Greater Asia for all of our products in the gut performance range, the microbials, the acidifiers, and the phytogenics. I have a PhD in animal nutrition, and while my background is in pigs, I've been working actively in the poultry and ruminant sectors as well, and happy to lend my expertise to today's webinar. Thank you very much, and I hope you enjoy it. Over to you, Gangga.
Thank you, Dr. Neil. So, good afternoon, and ladies and gentlemen. My name is Gangga Widyanugraha. So, my background is a veterinarian. I'm from Indonesia. I joined BIOMIN on 2015, and BIOMIN now also is part of DSM. So my role is a regional technical sales manager of poultry. So I'm supporting some countries like India, Indonesia, then Nepal, Sri Lanka, Myanmar, Indonesia, and as well as Vietnam and Philippines. Thank you for joining this webinar. So over to you, Dr. Selvam.
Thank you, Arun. Thank you, Dr. Neil. Thank you, Dr. Gangga. Good afternoon, friends. I am Dr. Selvam Ramasamy, Technical Director, BIOMIN India. So I have done my BBSc from Namakkal Veterinary College, followed by MBSc, which specializes in pharmacology and toxicology from Madras Veterinary College, Chennai. I have had over 15 years industrial experience covering both human as well as animal healthcare segments. My expertise includes new product development, conducting veterinary clinical trials in different target species of animals, and providing techno-commercial support to sales and marketing team. Thank you, Arun. Over to you, Arun.
Thank you, gentlemen. All three of you bring in such depth of knowledge and years of international experience to this webinar. Now let's move to today's agenda. We just want to share with the audience the key topics we are going to cover today. So as you can see in the screen, the first topic will be about feed hygiene, risks of feed contamination, and how and why acids are used to reduce such risks. A very important topic will be handled by Neil. Next one is about water hygiene and how to apply acids to eliminate gram-negative pathogens while supporting good bacteria. And of course, another important component in this presentation is the ideal pH of drinking water. This will be handled by Gangga. And the third session will be handled by Dr. Selvam, which is about gut health and how do the three main components of top 3 focus on pathogens in the gut while normalizing microbiota. So just to inform you all, we will also have a dedicated Q&A session at the end. So if you just see the right hand side corner of the screen, you will find a chat function. So you can just feel free to submit your questions by typing or keying in your questions. Another thing is about today's polls. To help the audience get the most out of this webinar, we just want to be interactive. And so we will also run a poll before each session and get the audience further involved. Now, let's start the first session with a quick poll. I hope you should be seeing the poll question and the options. So if you want me to read it, do you currently use something in the feed to assist in optimizing feed hygiene? So the options are number 1, no. Number 2, yes, an organic acid. Number 3, yes, a phytogenic. And number 4, yes, a mixture of products. So the audience are very active. Even before I finished reading, almost 50% have voted. I will just wait for a few more seconds and see how many are responding. The responses are coming in. Let me check whether we will reach the full attendees responses. Let's see. Okay, let me stop this now, poll now. So the response is 50% of the people, responses are telling us like, yes, they are using organic acid. And 30% they are saying like, yes, a mixture of products. And I'm sorry, 15% are saying no, and 4% are saying phytogenic. So I will now hand it over to Neil. He's the right person to, decipher and give more insights from these results. Dr. Neil, over to you, and let's get started with your session, please.
Thank you very much, Arun. Very interesting results there. Obviously, everybody's excited and already using acid, so hopefully I can still extend their knowledge in terms of the ability. But interesting, there's some people that also aren't using anything and or using a combination of products. So very interesting results, and thank you very much for your input there to the attendees. So moving into the presentation, what I would like to talk to you briefly today is, introduction to the enemy. What is the problem with feed and water hygiene? A quick refresher on the importance of gut health to animal performance. Bit more of a technical definition of what feed hygiene is, so at least we're all on the same page. And then I'd obviously like to introduce the role of organic acids in optimizing feed and water hygiene. And as Arun has mentioned, if you have any questions, please ask them at any time in the chat box, and we will endeavour to get them all addressed at the end of the seminar. So I guess just to put it in perspective, when we're talking about some of the microbiological risks in feed for animals that have an impact for humans, these are the main contenders: Clostridium, E. coli, Salmonella, Listeria, and Campylobacter. And so at the heart of all of our activities in raising food for raising animals for human food is we want to make sure that the safety of humans is paramount. But we don't also want to forget about the safety of our animals as well. And so this is just setting the scene in terms of what are the main components of, or the main antagonists in terms of feed hygiene. When we look at feed efficiency in animals, and we know that there's many things that influence it. There's the genetics, environment, housing, staff, the biosecurity, hygiene. So this is on-farm hygiene in terms of cleaning, how quickly and adequately it's done, and how the frequency. But we also know that the disease status, the types of diseases, the frequency of outbreaks, all have a big impact on feed efficiency. But where we're focusing on today is somewhat on the feeding side. And this can, the type of feed, the amount that the animals eat, the digestibility and availability of the nutrients, but more importantly for today's seminar, the ingredient quality. What is the microbiological levels of those ingredients? And all of these will have an impact. And so while we're focusing on feeding today, we have to be mindful that this is only one part of the holistic system. If we don't have all these other things right, all the activity we do in terms of making the best feed will not result in optimal performance. But why I've mentioned this to you today is that when we look at all these things that influence feed efficiency, we know that they also directly have an impact on gut health. And why are we worried about gut health? Well, the gastrointestinal tract, the GIT, is the most exposed surface. It's basically like the outside of the animal. Whatever the outside of the animal sees is what we also see in the GIT. And so there's constant exposure to all those potentially harmful substances and the microorganisms via the feed and the water. And the GIT is a very selective barrier. It has to combine the physical, the chemical, the immunological, as well as the microbiological barrier. And it has to be in balance so that it doesn't exclude all the nutrients, but it does have to exclude all of the toxins and the bacteria. So it's a very important organ that we need to take care of as best as we can. And we know that the development of the intestine is faster than the rest of the bird's body. So if you don't have the GIT being developed at the right rate, then the rest of the bird can't develop either. So we have to have optimal performance of the GIT development. And in terms of managing gut health, I believe it's more of a prevention rather than a cure approach. And it can be aligned with firefighting. If you get in early with a small hose and put out the fire, you're going to be much more successful at controlling, optimizing gut health. Manage gut health properly and early before it gets out of control. Because once you have raging dysbiosis, diarrhea, wet litter, etc., you're going to use and need to use more expensive control measures, and they may be less effective. And this is where we need to bring in the bigger fire truck, or in some cases come in with the aerial bomber, equivalent to antibiotics, and completely swamp the gut. And while we know that antibiotics are very detrimental to the pathogenic bacteria, they are also quite frequently detrimental to the beneficial bacteria. So we should reduce our use of antibiotics as much as we can. What happens when animals experience a bacterial challenge, and why are we interested in controlling it? The main thing is that the bacteria will compete for nutrients. They can also secrete toxic compounds, particularly endotoxins. And we know that endotoxins, and in collaboration with mycotoxins, have a very detrimental effect on the gut barrier, the gut integrity. So we need to reduce the amount of endotoxins being secreted by those gram-negative bacteria. We know that bacterial challenge will decrease the nutrient digestibility, and it also stimulates the turnover of the absorptive epithelial cells. And this is detrimental in two ways. One, you lose a lot of nutrients, and secondly, it takes 4 to 7 days for those epithelial cells to mature and become fully functional. So as you increase the turnover of the absorptive epithelial cells, you're going to limit nutrient availability and digestibility even further, because these epithelial cells are less functioning. And we also know that it increases the stimulation of mucus secretion. And while we do want some mucus secretion, the mucus provides an important lubricating barrier to the gut wall. We don't want too much, because too much mucus will prevent nutrient absorption. So we know that all of these things are quite severe challenge to the animal. And finally, bacteria will stimulate the inflammatory responses. And these inflammatory responses consume a lot of energy and amino acids, which will be diverted away from effective growth. And so the net result of all of this is that the bird will lose a lot of energy and nutrients because of this bacterial challenge. So what can we do about it? How do we manage it? Well, there's two main things that are important. One is to keep the pathogenic bacterial level low, both in the environment, because that ultimately impact what goes on in the gastrointestinal tract. And secondly, we need to optimize the animal's defense mechanisms. And it's a bit of a circular argument. If we can keep the environmental and gastrointestinal tract levels low, then you're not going to be impacting on the defense mechanisms. So both of these objectives can be achieved by optimizing step one. And from the gastrointestinal tract's perspective, we have to remember that pathogens can come from the feed, they can come from the water, or they can come from the environment. And if we manage the first two, if you can reduce the amount of pathogens coming in via the feed or water, then you're going to be reducing the shedding and therefore hopefully improve on the environment. And I deliberately put some breeders into this picture because we often focus on broilers, but breeders and layers are really important as well to be considered when we're talking about gut health. I mentioned at the start, I wanted to give you a bit of a technical, and this might be a bit heavy going for late in the afternoon, but technically feed hygiene is often defined along a similar viewpoint as the common EU definition. And the EU defines feed hygiene as the measures and conditions necessary to control hazards and to ensure fitness for animal consumption of a feed, taking into account its intended use. And so there's a whole lot of registration requirements, regulation requirements that exist in all countries around feed hygiene, and India is no different. And at the end of the day, it's all about protecting the humans that consume those animals. However, realistically, it is very difficult to control all the steps up until the point that an animal consumes a feed, because there's so many different areas where microbial contamination can occur. Therefore, often what happens is there's a critical control point that's placed on the feed mill. So the feed mill becomes the most important part, or one of the most important parts, for reducing microbial contamination and being the chief determinant of feed hygiene. But you have to also remember that recontamination of the clean feed downstream of the feed mill is still possible, and this needs to be managed, because this could happen in your storage on farm, in the transport of that feed, any step along the way. And although I talk about feed hygiene, we don't want to forget about water hygiene. And this is a very separate, but still very complementary to feed hygiene, and should not be ignored. In terms of where does the microbial contamination come from? Well, there's many and varied places. So the microbial contamination can start in the field as the crop is growing, and often this can happen because birds and other animals are contaminating the crop. It can also continue during the various transport and storage stages of the grains. If the transport trucks, the bulk trucks, haven't been cleaned properly, or were used to carry, manure perhaps in some countries before you put your grain in, then this is obviously a very critical source of contamination. But the microbial contamination can also occur in other products other than grains. And this is particularly relevant when we start talking about byproducts in the production, the storage and transport of byproducts. And these are things such as your dried distillers grains, as well as soybean meal, remembering that soybean meal is actually a byproduct of the soy oil production process. So there's many areas where microbial contamination can come into it. And particularly for those people that are using animal byproducts, this is in particular concern because of the rich environment that the animal byproducts make for bacteria. And we also know that water hygiene can fluctuate dramatically, both over the short and long term, particularly if you're using wells or open dams where birds and other wildlife can come in and contaminate them. And depending on the wet season or the dry season, the actual levels can fluctuate quite dramatically over both the short and long term. And so awareness of these changes need to be observed. So why do we use organic acids to aid in feed and water hygiene? Well, there's quite a number of valid reasons. Organic acids offer a unique strategy to control microbial contamination because they can be applied in either liquid or powder form. So liquid makes it very easy to preserve raw materials. Powder form makes it very easy to help with finished feeds. They can be applied multiple times in the ingredient supply chain if required. So for example, in a high risk ingredient, such as a dried distiller's grain or some other more moist component, you can apply a liquid acid to that ingredient, and you can then apply a powdered product to the finished feed when you're making the finished feed to protect it from all of the risks. We know that organic acids are long acting, so they will provide extended protection in the feed. They don't evaporate, they don't disappear very quickly. We know that also that organic acids are easily activated by the moisture in the feed and air. So you don't have to have a liquid environment to really, in terms of the feed, to make them activated. There is enough moisture in the feed to actually identify that or make that protection within the close proximity to the acid molecules. We know that acids are both very effective and very economical, and more importantly, they're safe to use for both humans and animals. And particularly, we know that acids don't induce antibiotic resistance. And in fact, some of the research that BIOMIN's been doing has shown that in the presence of products such as top 3 that you will hear about later, we can actually reduce the number of antibiotic resistance genes when feeding the acid product top 3. So many good reasons, I believe, for why using an organic acid for feed and water hygiene. So in terms of what products do we want to recommend? Well, BIOMIN has a range of products. For feed hygiene, we would recommend a product called Biotronic Clean Feed. And this is particularly for feed preservation over long periods of time. If you just want feed hygiene and initial gut health, the recommended product in this case would be Biotronic SE. And SE is often abbreviated for Salmonella and Enteritidis, Salmonella and E. coli. In terms of really strong gut health control, and you will hear about this in Dr. Selvam's presentation, the recommended product to use is Biotronic Top 3. And so we would say that if you wanted a little bit of feed hygiene and a little bit of gut health, the best product combination would be to use, say, 1 kilo of SE and 1 kilo of Top 3. And as Dr. Gangga will talk about, the most effective product for water hygiene is a Biotronic Top Liquid product. So as you can see, there's a number of products in the BIOMIN portfolio that will address your feed and water hygiene needs. So please reach out to your local BIOMIN representative to get more information on these products and their application rates. So in conclusion, Mr. Chairman, I hope I've identified quite briefly though, that poor feed and water hygiene can have a significant impact on animal performance. More importantly, bacterial contamination of animal products can severely affect the consumers of those products. And this is why we want to be very clear about how we position the products and the advantages for our animal producers. We know that organic acids provide a cost-effective method for optimizing gut health. And hopefully now my colleagues will give you more details on how organic acids in water and feed can be an important part of your health management strategies. And with that, I will hand it back to you, Arun, for your next poll question.
Thank you, Dr. Neil. Appreciate that. I already see in the chat box some specific questions for you.
Excellent.
Already. And it was very informative listening to you. And I would also like to assure the audience that, just in case you missed some key points for some reasons, we got you covered, as we will be sending the recording of this webinar when it is available to your registered email addresses. So before I hand over to Gangga, I'm going to run the second poll to get the insights from the audience. Your responses are really of great value to our presenters and as well as to BIOMIN as a whole. So now let me run the second poll question. So you should be seeing the question in the screen and the options for that. What's the pH of drinking water in your farm? Sounds simple, but our Gangga will go into great details on this. Above 8, 6 to 8, 4 to 6, below 4, and the final one is not sure. Great response from the audience. We got 3/4 reply already. I will wait for a few more seconds before I close. This is going to be really interesting for the next presentation, because Gangga really works in this area. Great. Just give you 5 more seconds. And okay, let me close this now. So I will share the results. You are seeing the results on the screen, Gangga, for your special information too. What's the pH of drinking water in your farm? Above 8, 6%, 6 to 8, 61%, 4 to 6, 29%, below 4, 2%, and 3% are not sure, which is quite okay. So, Dr. Gangga, you are the expert in this topic, and please let us know.
Yeah.
What do you think about this? Over to you.
It's nice result, Arun. I think based on my experience, I visited in India to many farms, and I also checked some initial drinking water pH on the farm, and yeah, it was mostly from 6 to 8 pH. So I think it represents India, yeah, the polling. So I think some place I saw more than 8. Yeah, and also in the result, in the polling result, also showing some places are more than 8. So let me talk about this later. Yeah. So how to address this range of the water pH. So let me start. Okay. Yeah. So again, thank you everybody for joining this webinar. Yeah. So my topic today is about what's the ideal pH of drinking water and how to apply acids to eliminate gram-negative pathogens while supporting good bacteria. Yeah. So we know, yeah, we all know that water is the most important nutrient and has a lot of functions. I think if I ask you impromptu, so you can mention many things about the benefits of water. It's countless and limitless. But when we talk about in poultry, poultry consume approximately 2 times as much water on a weight basis as feed. Can you imagine? So it seems, poultry consume water more weight basis compared to feed. So that's why we need to really take care of this nutrient. Yeah. So water is not just only for drink. So many benefits. Yeah. So in poultry, water can be used for softening feed in crop. Yeah. And then why softening feed in crop? Because birds have limited saliva, so they need more water. Yeah. Then carries feed through digestive tract, so it is very essential for digestive process. And then water also necessary for reducing air temperature. Yeah, and it actually includes like evaporative cooling pad and as well as some fogging system. Yeah, many things. Yeah. Then water is also important because it's a key component of blood. Yeah, we all know that. And also this is essential for growth and reproduction. Yeah, we know that in meat contains 70 to 75%. It contains water. Yeah, eggs around is around 74% contain water. Yeah. So I put on the board mark. Yeah. So a drop in the water consumption is often the first indicator of flock problem. I think we all agree about this. Yeah? So when we see the reduction of the water intake based on the daily report, so you can suspect many possibility of the problem. Yeah. So that's why, yeah, water is very important, and especially this is often neglected nutrient. Yeah. We need to consider about, especially about pH. Why? Next, not only many benefits, water also carries many pathogens. Yeah. So this is water is very important role in transmission of some bacterial diseases such as like this Mycoplasma, and then E. coli, Pasteurella, as well as Salmonella, and then some viral diseases as well as protozoan diseases such as the coccidiosis and histomoniasis. Yeah, imagine, 2 types of feed will carry many of these. So that's why we need to really careful on the water. Yeah, and here's the example what will happen if we don't take care properly of the water. Sometimes we can see some lesions like this. Yeah, We could see some fibrinous perihepatitis in liver or pericarditis in the pericardium, and sometimes we can find some cheesy material, or exudates. So most people think that this is probably caused by E. coli. They're probably right. But for myself, it's not the one and the only E. coli can cause this. Yeah, some other gram-negative pathogenic bacteria also may contribute some incidents like the cheesy exudates and egg peritonitis, for instance, in the long-living birds or laying hen, in layer and breeder. So this slide showing some gram-negative pathogens in poultry, not only E. coli, not only Salmonella. You can see also some possibility of Campylobacter, then Pasturella, Mycoplasma, Yersinia, Vibrio, and as well as Pseudomonas. And if we see on the third column, mostly these pathogenic bacteria are not growing on pH 4.5 and below. So that's why it's very important to make water on the pH 4.5 to stop the growth of the pathogenic bacteria. I think you all agree with this. Now if we see this graph, you see this is a graph of the sample of E. coli and Salmonella and Clostridium perfringens and Campylobacter jejuni. So we see that the pH tolerance of this bacteria, we see the optimal growth is around from 4 to 8. Let's say it's around about 4.5 to 8. So in this range, all of these pathogens are very, very thriving. More than 4.5. Okay. And in the other hand, pH tolerance of beneficial bacteria or good bacteria, they are showing in the range pH from 4 to 5 their optimum growth. So by targeting the pH water on 4.5, not only stop the pathogenic bacteria, but also it will promote the growth of the beneficial bacteria. In this graph, it's showing a sample from good bacteria, isolated from chicken's gut. We call it a probiotic. You can see Enterococcus faecium, Pediococcus acidilactici, Lactobacillus salivarius, Lactobacillus reuteri, and Bifidobacterium animalis. These are the most beneficial bacteria, especially for poultry. So less than 4.5, pathogens are suffering, but it will bring some mutualistic relationship among the beneficial bacteria or probiotics with acidifier. Then if we talk about the ammonia in acidic water, ammonia is less toxic in an acidic condition when the pH is low. At pH below 7 virtually, all the ammonia will exist as ammonium ions, which is non-toxic. And at pH less than 6, the proportion of ammonia is very, very low, but the ammonium ions is very, very high. So by giving a liquid acidifier in water, it also will reduce the proportion of ammonia. Then talk about hardness, which is primarily a measure of calcium and magnesium contents, and for calcium, primarily measured by like calcium carbonate. This is also another problem of water quality in poultry production. Actually, birds are tolerant up to 1000 ppm hardness, but this could have an impact on mineral deposition on farm equipment and drinking water pipes, causing a rapid buildup of a lime scale and biofilm. And hard water also affects the solubility of powder medications. Sometimes it is hard to mix a medicine. For example, like anti-coxy medicines. Like let me give an example, like tortrazuril. For instance, or maybe some antibiotics like chlorphenicol. Yeah, it's hard to mix medicine of that in a high hardness. So this is also a part of water that we need to consider also. Yeah. So next one, the important part of the water. Yeah, we have to flushing. Yeah, of the water line. So this is the ideal cleaning procedure of the water line before using it for poultry drinking water. Yeah. So proper cleaner and leaving long enough time are the key of success of flushing. Yeah, and cleaning the whole system is essential, including water lines, standpipes, and maybe regulator and distribution pipes. And not only that, we also need to evaluate the efficacy of a product. Yeah, I mean the product for flushing by collecting swabs before and after cleaning. And for the final flushing, we need to give final flushing, final spray with sanitizers, because it will help to kill any bugs that survive from line cleaning. Yeah, during the flushing. Yeah. So this is the example of the chemical for water sanitation. Yeah, for flushing. It's a hydrogen peroxide. Yeah, H2O2. Mostly we give the dose about 15 to 20 mg per liter water. Yeah, and then we need to excuse me. We need to leave it for around 2 to 3 hours and then flush it. So it can be used for oxidizing iron and manganese. Yeah, but the weakness is it can be dangerous to store and handle because it's flammable. That's why it's not commonly used alone due to its flammable nature. But anyway, it's a recommended chemical for flushing. Yeah, hydrogen peroxide. Next is chlorine. Yeah, with a dose of 2 to 5 ppm. Yeah, sometimes using 3 ppm commonly. With 3 ppm active chlorine. This is also can be used for water sanitation. So you just need to prepare 4 to 6% of solution and then dilute the 1 liter hypochlorite with 10,000 liter water. Yeah, but this is affected. This is also depends on the pH. Yeah, but for applying this, the best pH around 4 to 7. Yeah, and also water temperature will decrease the efficacy of chlorine if it is above the 20 degrees of Celsius. Yeah, and this is really also another mutualistic symbiotic. Yeah, acidifier and chlorination. Yeah, for poultry drinking water. Yeah, liquid acidifier and chlorination have a mutualistic relationship. Yeah, why? Because if chlorination applies in acidic water, it becomes hypochloric acid, which is a stronger disinfectant and oxidant. But if it applies on alkaline water, it will just become hypochlorous ion. Yeah, this is the explanation. So hypochloric acid, yeah, is 80 to 300 times more effective as a sanitizer than hypochlorous ion. Yeah, and free chlorine not considered effective unless it is 85% contains hypochloric acid. Okay, and the other chemical. This is organic acid. Yeah. So organic acid also can be used for water sanitation. So it will kill bacteria and then enhance the efficacy of chlorination. Yeah, surely. Yeah, as I mentioned in some slides before. And then it will increase the digestibility protein. Yeah, by reducing the buffering of the feed passing from the crop to prevent regulus. And this acidity also plays a major factor in the transformation of pepsinogen to pepsin. Yeah. Then lowering ammonia level in litter, surely. Yeah. as I explained before, and the combination of organic acid is important to optimize control of pathogenic bacteria. Yeah. So the take-home message, from my presentation, that the liquid acidifier, which target to pH 4.5, will reduce the challenge of gram-negative pathogenic bacteria in drinking water, and it will promote beneficial bacteria and also increases effectivity of chlorination, and last but not least, it will result in less toxicity of ammonia. Over to you back, Arun. Thank you very much for giving me a time.
Okay, that was really a good one, Ganga, with a lot of examples from the field, and I'm sure that many from India could connect with the examples you provided. So
Thank you.
now, yeah, thank you. And of course, Ganga should be a familiar face to most of you. Most of you should have seen him on the field before this COVID. Now, let me run the third and final poll question, which will also provide a much needed talking point to the final or the third presenter, Dr. Selvam. I'm running the poll now. Please feel free to choose the best. What do you use to prevent pathogenic E. coli escaping from the poultry gut and infecting other organs? What do you use to prevent pathogenic E. coli escaping from the poultry gut and infecting other organs? Acidifiers in feed, acidifiers in drinking water and feed, combination of phytogenics, probiotics and acidifiers. And the last option is antibiotics or AGPs, antibiotic growth promoters. I'll wait for a few more seconds. Responses are coming in. I'm glad that most attendees want to be interactive and engaging with these polls. I'll take few more seconds before I close. Okay, 3/4 have keyed in their responses. Now I close the poll. You should be seeing the results in the screen. Combination of phytogenics, probiotics and acidifiers. I think that's great, but I leave it to the expert to comment on that. Acidifiers in drinking water and feed 30%, antibiotics or AGPs 11%, and acidifiers in feed 6%. So with that, I would like to hand over to Dr. Selvam, who will go into this poll and he will give you his opinion and move on to his last and final presentation. Over to you, Dr. Selvam.
Thank you, Arun. Hi, Arun. It's a very interesting note that majority of the people have opted for the options, combination of acidifiers, phytochemicals, and probiotics in feeds. Yes, acidifiers alone give protection against gram-negative bacteria.
Selvam, you can open your presentation.
Acidifiers alone will give protection against gram-negative bacteria. When acidifiers are combined with phytochemicals, it will enhance the antimicrobial spectrum of organic acids covering both gram-negative as well as gram-positive bacteria. This inclusion of probiotics ensures quicker establishment of gastrointestinal beneficial microbiota. So combination of all three will be most complete and comprehensive solution in terms of providing best gut health for the broilers and in turn improving the performance of the broiler birds. And also it can effectively replace the in-feed antibiotics and also avoid the antimicrobial resistance. Thank you. So let us see my session. Okay, my presentation covers the Biotronic top 3, the flagship brand, acidifier brand from BIOMIN, and how does each component of Biotronic impact the gut health, followed by a couple of field trial data wherein we have demonstrated efficacy of Biotronic top 3 in target species. Biotronic top 3 contains 3 major components. The first component is a synergistically acting blend of organic acids. The second component is phytochemical constituent cinnamaldehyde. The third and most important constituent is BIOMIN permeabilizing complex. This BIOMIN permeabilizing complex is unique and innovative substance which disrupt the outer cell membrane of gram-negative and gram-positive bacteria, thereby facilitating entry of organic acids and phytochemical constituents into the cells, enhancing their antimicrobial spectrum. The fourth constituent is sequential release medium. It's a vermiculite because it's as a functional carrier. All these three components are blended onto this vermiculite. So this vermiculite ensures slow and sustained release of these three active components throughout the gastrointestinal tract of broilers and chickens. So let us see the mechanism of action of individual component. The blend of organic acids. Our product Biotronic Top 3 contains 3 acids: formic acid, acetic acid, propionic acid, and the salts in the proportion of 4:2:1. So this is a mode of action of organic acid general. In general, organic acids are weak carboxylic acids, which means they partially dissociate into their respective ions, that is hydrogen ions and RCCOH minus anions. These hydrogen ions interact with electrons on the molecules of water and forms hydronium ions. This hydrogen or hydronium ion is responsible for reducing the pH of the environment, like feed, water, and just of the GI tract. This acidic environment makes the under acidic environment, these pathogenic bacteria are not able to survive or thrive. As a result, their growth and multiplication is arrested. When acids are in undissociated form, they exhibit antimicrobial effect. This undissociated form of acid permeates easily through the bacterial cell membrane through passive diffusion. Once they are inside the bacteria, they dissociate into respective ions, hydrogen ions and RCOH ions. It produces an antibacterial effect. However, their effect in pH reduction and antimicrobial effect varies depending on their pKa value. This is the table showing pKa values of commonly used organic acids. The pKa is the pH at which 50% of acids remains in their dissociated form, 50% acids remain in their undissociated form. That means the concentrations of dissociated and undissociated form of acids are equal at this pH. So higher the pKa value, better would be the undissociation. Lower the pKa value, the acids are readily able to donate the hydrogen donors for pH reduction. So higher the undissociated form, it exhibits antimicrobial activity. Lower the pKa value, it exhibits higher pH reduction in the environment. So what will happen when pH reduction happen in the environment? So as I mentioned earlier, as I already covered by Dr. Neil, this acidic environment is a barrier to bacterial entry, especially acid-sensitive bacteria like E. coli, Salmonella, Vibrio, and Campylobacter. They are all sensitive to acidic environment. In the sense, they are not able to grow and multiply under acidic environment. As a result, their growth is inhibited. Whereas the beneficial bacteria like Lactobacillus species and Bifidobacter species, they are able to survive under acidic environment. They will grow, proliferate, and increase their colonization in the gastrointestinal tract. The second function is the reduction in pH activates the secretion of gastric and pancreatic enzymes like pepsinogen, trypsinogen, and chymotrypsinogen will be activated into a respective active form like pepsin, trypsin, chymotrypsin. They facilitate digestibility of protein in the gastrointestinal tract. And also low pH also activate the secretion of amylase and lipase from pancreas. They will improve the digestion of carbohydrate and lipids respectively. The next result is better gut health, improved digestion, absorption and utilization of nutrients, that results in better performance of birds. So how does the organic acids exhibit their antimicrobial effect? Undissociated form permeates easily through the outer microbial membrane. Once they enter inside the cell, they will be dissociated into hydrogen ions. This hydrogen ion is detrimental. As a defensive mechanism, bacteria will try to pump out these hydrogen ions by using H+ ATPase pump. As more and more acids are getting accumulated inside the bacteria, so more and more energy is deployed towards pumping or expelling the hydrogen ions outside the membrane by the bacteria. In this process, the bacteria will use most of its energy for pumping the hydrogen ions out. So very less energy is available for metabolic functions of bacteria. As a result, there will be inhibition of protein and DNA synthesis in the bacteria that leads to bactericidal effect. So this is how organic acid exhibits their antimicrobial effect on the bacteria, pathogenic bacteria. So we have done a in vitro studies wherein we have demonstrated antimicrobial effect of individual organic acids that is present in our product Biotonic Top 3. So as you see this graph, so we have tested all common organic acids. So we have found formic acid, acetic acid, and propionic acid. They exhibited better inhibitory effect on the common Salmonella pathogens like Salmonella typhimurium and Salmonella enteritidis. We have also evaluated individual organic acids against E. coli strains, common E. coli strains of O55, K59, and O128H. So here also we have obtained the same results. Formic acid, acetic acid, and propionic acid, they have shown better inhibitory effect on E. coli strains when compared to other organic acids. The second component present in Biotonic Top 3 is the cinnamaldehyde. It's a bioactive organic compound obtained from cinnamon bark of cinnamon trees. It gives characteristic odor and flavor to cinnamon. So before seeing the mode of action of cinnamaldehyde, we should know how does the cell division happens in pathogenic bacteria. So FITZ protein, this protein is very essential for cell division in pathogenic bacteria like E. coli, Salmonella, Campylobacter. So this protein is the first protein moving to the site of cell division. This FITZ protein is encoded by a gene FITZ. The FITZ protein assembles into protofilaments. These filaments forms a septum. These filaments form a septum on the longitudinal midpoint of the dividing cells. These FITZ proteins also recruit other proteins, and it provides a cytoskeleton scaffold to this Z ring. So FITZ, Z ring is the site of cell division. The Z ring has the ability to constrict on its own. When these protofilaments are pulled on each other, it will constrict in the midpoint. It divides the cells into two. So this is how cell division happens in pathogenic bacteria. But cinnamaldehyde is known to inhibit this FITZ protein as well as its assembly into Z ring. As a result, the polymerization of FITZ protein into protofilaments is prevented. Z ring formation is prevented. So cell division in the pathogenic bacteria is arrested. So this is how cinnamaldehyde exhibits its antimicrobial effect by preventing cell division and arresting of Z ring formation. Cinnamaldehyde also is known to activate the endogenous digestive enzymes. So that will improve digestion, absorption, and utilization of nutrients like carbohydrates, proteins, and like lipids. Cinnamaldehyde also able to inhibit quorum sensing in pathogenic bacteria. The quorum sensing is a mechanism by which bacteria communicate with each other. Once this quorum sensing, this communication signal is prevented by cinnamaldehyde, there will not be any more communication between pathogenic bacteria. As a result, the growth and multiplication of the bacteria will be prevented. Cinnamon is also known to increase the abundance of beneficial microbiota, that is lactobacillus in GI tract of birds. At the same time, they are known to reduce the abundance of pathogenic bacteria like Clostridials and Enterococcus. So that will reduce the incidence of necrotic enteritis and coccidiosis in chickens. So when organic acids are combined together with phytochemicals, they act synergistically with each other and increase their spectrum of activity. So when they are combined together, they are known to have broad spectrum antimicrobial effect covering both gram-negative and gram-positive bacteria and providing better gut health, improving the absorption, digestion and utilization of nutrients, resulting in better performance in chicken. So another third component is a very unique and innovative substance, that is BIOMIN permeabilizing complex. This permeabilizing complex is known to weaken the gram-negative bacteria outer cell membrane. So once the cell membrane is weakened or damaged, it facilitates entry of organic acids and cinnamaldehyde inside the bacterial cell membrane, so thereby increasing the antimicrobial effect of organic acids and cinnamaldehyde. So this is a study we have done. It's a fluorescent probe NPN assay, wherein we have demonstrated the effect of permeabilizing complex on bacterial cell membranes. The NPN is a hydrophobic dye. When bacteria is intact, normal, when bacterial cell membrane is normal, it does not allow entry of this dye inside the cell. Once damaged, the bacterial cell membrane allows entry of NPN cells that will give a prominent fluorescence under fluorometer. So this is a study wherein we have demonstrated the efficacy of this product. So we have used EDTA as a reference compound, and we have tested permeabilizing complex at different concentration levels: 0.001%, 0.002%, and 0.003%. Higher the fluorescence, RF units, its reference relative fluorescence units. Higher the relative fluorescence units, higher the damage to the outer cell membranes. So this graph clearly indicates that when permeabilizing complex is added, it is able to produce more damage to the outer cell membranes of the pathogenic bacteria when compared to control and reference standards, EDTA. So blue one is E. coli, the orange one is Salmonella typhimurium strains. We have also done a growth inhibition test, wherein we have measured susceptibility and growth of different E. coli and Salmonella strains. So this is a culture study. We have kept a control. This one is control. The blue is control. The red one, orange one is antimicrobial mixer. The red one is only BIOMIN permeabilizing complex alone. This blue line is antimicrobial mixer plus BIOMIN permeabilizing complex. This is Salmonella typhimurium strain inhibition test. So as you see in the graph, when we have added BIOMIN permeabilizing complex alone, there is no difference observed between control and BIOMIN permeabilizing complex alone. Whereas antimicrobial mixer, okay, they are able to reduce the OD of culture medium, but there is no sustained and consistent reduction in the OD of the culture medium. When we combined antimicrobial mixer along with BIOMIN permeabilizing complex, there is no OD, no turbidity of the culture medium at all. That means there is no growth of bacteria at all. So bacterial growth was completely arrested when BIOMIN permeabilizing complex was added along with antimicrobial mixer. So this is growth inhibition of E. coli strain. So here also we have obtained almost similar results. When BIOMIN permeabilizing complex was added along with antimicrobial mixer, there is no turbidity. That means there is no growth of any bacteria at all in the culture medium. So the fourth component is carrier. we call it sequential release medium. It's a vermiculite. It's very easy to handle, waterless, and mold resistant. This vermiculite, when heat processed, it expands into box-like pellets composed of multiple layer of thin plates. These active compounds are adsorbed onto these thin plates. When ingested by the birds, it ensures slow, gradual, uniform, and sustained release of active components throughout the GI tract. It's not just a carrier; it's a functional carrier. So my next session, my next part covers some in vivo trials that we have done to demonstrate efficacy of the products in poultry. So this is the BRALE trial we have done at Nanjing Agricultural University, China. So here there are two treatments: standard diet. The second one is standard diet plus biotronic top 3, 1 kg per ton of feed. The trial was done for 42 days. Total number of birds in each treatment is 200 birds per treatment. Each treatment have 4 replicates containing 50 birds each. So we have evaluated impact on cecal microbiota. As you see the slides, there is significant reduction in pathogenic bacteria like E. coli. Normal control, it was 8.45, whereas in treatment groups 7.84. There was a significant reduction in E. coli count as well as salmonella and Clostridium perfringens counts when the birds are treated with biotronic top 3, 1 kg per ton of feed. But it doesn't decrease this lactobacillus count, whereas the count of lactobacillus was increased significantly. It has gone up from 8.2 to 9.1. So it clearly shows that the biotronic top 1 has negative impact on pathogenic bacteria, but at the same time it's sparing effect on positive beneficial bacteria like lactobacillus. So another trial we have done at Ankara University, Turkey. So here also there are 2 treatments with 8 replicates pen. Each replicates having 14 birds per pen. So here there are 2 groups. One is control. So another group is treatment group, biotronic 1 kg per ton of feed. Trial was done for 42 days. So this is the results of the trial. So there is about 1.7% improvement in final body weight, 1.4% improvement in feed intake. The significant feature is there is about 32% reduction in mortality rate in treated group. Also, there is about 2 points improvement in FCR when compared to control group. So we have gotten ROI of 1.9:1. So in the same trial, we have done a histology analysis of intestine. So this is treated group. This is a control group. In treated group, there is about 28% improvement in villus length, but whereas crit death was significantly reduced by 25%. So let us see the morphometrical analysis. So this is the morphometrical analysis of intestine. Villus height is improved by 28%. The crit death is decreased. 176 in control birds become 132 in treatment group. Also, thickness of intestine is also reduced from 142 to 112. So organic acid our product have impact on thinning of intestine. Thinning of intestinal lines always increases the absorptive surface, whereby improving the absorption of nutrients from the intestine. So when we have calculated villus height to crit death, so there is significant increase in villus height to death ratio in case of biotronic top 3 treated group. In normal it was 7.82, it has become 13.79 in biotronic top 3 treated group. So this is another trial we have done in layer birds in France. So here there are two treatments with 252 birds per treatment, 14 replicates containing 36 birds each. There are standard diet, then standard diet plus biotronic top 3, 1 kg per ton of feed. This trial was done between 40 and 70 weeks of age. The results is represented in this slide. So there is about 1.2% improvement in laying rate percentage, and cumulative X is 1.3. Average egg weight is also, there is no much difference in average egg weight between control and treatment group. Whereas cumulative egg mass was increased by 1.1%. Viability per week is also increased in case of a treatment group. The 98% in control group become 99.2% in treatment group. So it clearly shows that biotonic top 3 inclusion both in broilers as well as layers have positive impact on the performance of the birds. Thank you so much, friends, for your kind attention.
Okay, thank you, Dr. Selvam. We really apologize because we are taken 5 more minutes of your time, attendees, but we are going to compensate that by answering many questions, because only this session we really note, like, lot of questions have come in. So we are going to address few questions now, and the rest of the questions we will be addressing in future. Like, we will be sending you the responses in your email IDs, or our BIOMIN representatives will contact you. So the first question I would just like to ask Neil. It's a question from Manoj Chakraborty. Dr. Neil, the question is, I use apple cider vinegar. Is that an organic acid? What do you think?
Yes, absolutely. Nice question. Acetic acid is the acid in vinegar. So yes, it is an organic acid. And in fact, in some of the products in the BIOMIN, we actually include acetic acid. So yes, it is an acetic acid. Yes, it is an organic acid. The problem, though, is that when you use apple cider vinegar, you don't, unless you're doing good quality control, you don't actually know the concentration. And the problem with then using any acid in the water, if it's too dilute, acids are actually a feed source for bacteria. So just adding an amount of vinegar to the water line may not decrease bacteria, it might actually stimulate increase in bacteria. The other concern with using more concentrated forms of vinegar, if you go to a pure, is that the pH is somewhere between 2 and 3 of acetic acid. And if you use too much vinegar, you can actually turn the water intake of the birds off because it's just too powerful. And at pH 2 or 3, if you drop the pH, as Gangga said in his presentation, the ideal is around 4.5. So ideally, you need to use a buffered product, such as obviously what we're talking about here today, Top Liquid, to really have better control. But in rudimentary form, and if you take some control, yeah, apple cider vinegar is an organic acid and can be used, but there are some limitations with it.
I hope that answers your question, Mr. Manoj. So just now you touched upon pH. So the next question is indeed about pH, and it goes to Gangga. The question is from Khalid Diwan. What about consumption of water with pH 4.5? Gangga, this is for you. Gangga, no worries.
Just while Ganga's getting his sound, I'll quickly address that. There's no problem with drinking something at pH 4.5 because most of the liquids that humans drink, coffee in particular, is lower than pH 4.5. You add a bit of citric acid to your orange juice, your citric acid, it's already pH 4.5. So a little bit of zing to the water, actually, one of the advantages of drinking acidified water is that you actually increase your water intake and therefore your feed intake because the water is more palatable. It's got a bit of a zing. Personally, I prefer drinking a bit of water with lemon juice in it because it adds a little bit more zest to the water. So even though it sounds like it's really acidic, pH 4.5 is actually quite common, with things like wine, coffee, beer, soda. Coffee, cola is probably even lower than that. So there's no issue with drinking something that sounds like it's pH 4.5.
Great. Thanks a lot, Dr. Neil. And Dr. Neil, for your information, lot of questions have come in and which is most relevant for you. So we will be forwarding the questions to you, Dr.
Perfect.
Neil. Yeah. And now I'll just choose one last question. So this is a question from S. Kaka. What should be ideal combination of organic acids in water acidifier? Dr. Gangga, would you like to take it or did you hear?
Yes.
Yes, please.
So can you repeat one more time for the questions?
Sure. What should be ideal combination of organic acids in water acidifier?
Yeah, so the good combination, yeah, I think it already mentioned by Dr. Selvam about what's the best combination of organic acid, which is formic acid, propionic acid, and acetic acid is the best 3 acids among others for inhibiting the pathogenic bacteria, and especially in the product BioMIN top liquid also contains those 3 acids. And not only that, why you need to put just only on the pH 4.5 by using top liquid, because it has like a permeabilizing complex. Yeah? So we don't necessary to reduce more pH by using those acid more, especially up to less than pH 4. It is not necessary by getting on 4.5 with combination of formic, propionic, and acetic plus permeabilizing complex also will help to reduce or inhibit many of pathogenic bacteria.
And just if I could add, Arun, just quickly, one of the advantages is to actually use a mixture, and as Gangga identified a couple of different acids there, but it's more importantly, given the range of pH across the gut and the things that Dr. Selvam talked about in terms of, antimicrobial versus bacteriostatic, is we want to have a range of acids that will perform across an extended period of the gut to make sure you have the biggest effect. So that's why a choice of acids in a mixture is important.
There you go, Dr. Neil. Thanks for your comments. So with that, we have come to a close. And as you can see in the screen, we are also in the social media. And if you are more of a Twitter person, we have also a Twitter social handle in the name BIOMIN India. If you have any questions, please feel free to contact us through this social media handles. We also have a WhatsApp number linked to the Facebook. So please feel free. And I assure you that we will get back to you with all the responses for your questions. And on behalf of BIOMIN, I thank you all for listening. Of course, we are a little bit more than the scheduled time. We will compensate that in the future. So I appreciate the efforts of everyone and the time of the participants, and special thanks to the speakers and to the Asia Pacific marketing team for their support. So bye for now. Stay safe and take care, gentlemen, ladies. Thank you.
Thank you, Arun.
Thank you, everybody. Thank you, Arun.